Okay, so it's a great pleasure to introduce Jeff Kaye. I've known Jeff for about a century, or no, a long, long time. We sort of tangentially bumped into each other as neurology residents way back when. Jeff has had a wonderful career and trajectory in research on aging and dementia. He is currently a professor in the Department of Neurology at Oregon Health and Sciences University, and he is the director of the NIA-funded Alzheimer's Disease Research Center there. In recent years, Jeff has developed some pioneering approaches to using technology to identify information about how people are doing in all kinds of different activities, and has developed ways to collect this data in people's own homes using minimally obtrusive or intrusive methods. And I'm standing by describing any more details of that, because I'm sure he will do a wonderful job. So again, great pleasure to introduce Jeff.
Thank you very much, Doug. And it's really great to be here with you virtually. Of course, I wish I could be there with you personally. I've always had a wonderful relationship with so many people in San Diego. Well, looking forward to being able to come down in person sooner than later. I think we keep all wishing that things would clear up more definitively. In any event, great to be here. I'm going to share my screen. So as Doug kindly introduced me, I will be talking about these digital technologies and framing this mostly, though, in the context of clinical trials and trying to develop some better treatments for Alzheimer's disease and related disorders. And I'd like to start this by moving my slides. Let's see. There we go. So as we all know, there's been some terrible difficulties over many years in trying to develop what I would call meaningful treatments. This ad is about a decade old from the Alzheimer's Association. Despite all the talk recently, I think we're unfortunately still in the same boat of still in search of effective, meaningful treatments. And the history of this, I think this audience knows well. This is a graphic from an article that was published or reviewed actually now more than 5 years ago. But it's still the same sad truth that the top of the funnel, that is the number of treatments that enter the development, is relatively large, but by the time you get to the bottom, particularly in Alzheimer's disease, if you just compare that to the industry standard or average, is very low, and in fact is probably even lower now, given the 5 years that passed since this, 5 or 6 years since this graphic was made. It's not for lack of trying. This is the diagram from what's become now Jeff Cummings' annual report on drug development. Most of the drugs now are considered to be disease modifying, about 80%, with still some symptomatic treatments. But in general, there's not a shortage of possible treatments, but these are expensive and difficult, and unfortunately still haven't been successful. I think there's a lot of reasons for this. Two major ones is, I think we're still not sure about what the cause of a disease is, or disease is. I mean, we have this discussion about the heterogeneity of Alzheimer's disease. So it's hard to develop a treatment when you're not sure about what the target or targets are. The second reason, I think, is what I'm going to focus on today, and that is I'm not sure we know what success looks like. I'm not sure we have really agreed upon what are the measures of meaningful change. I think it's a little bit like climate change used to be, but we still have a lot of ways to go, I think, to really determine what we consider to be knowing what meaningful change is. This is really highlighted very starkly recently. So these are two articles actually that were published prior to the FDA announcements. On the left is one view. I'm trying to not make this political. So where the title says, aducanumab produced a clinically meaningful benefit. And then on the right side, the title is failure to demonstrate efficacy. And so there's clearly some confusion here. And so why might that be? Well, it's the same confusion as to why sometimes these slides are advancing and sometimes they aren't. Hello. There we go. Okay. So I think if you consider the way we assess people to determine whether there's a change in a trial, the tools we've been using for a long time, we know of high variability and both intra-individually and inter-individually or across individuals. And so in the bottom panel are ADNI data looking at the memory Z-score across normals, MCI, and Alzheimer cases. These spaghetti plots clearly show that variability. On the right is a panel from a South Korean longitudinal study looking at the CDR in people with suggestive cognitive impairment, amnestic MCI, and early Alzheimer's disease. Same principle is really demonstrated here. And underlying all this is there is actually probably high biological variability. So this is looking at the Florida-Peer SUVR scores over time. This is Bill Jagus and Landau's recent article looking at the dynamics over time. And so these are clearly challenges of high variability, but also the tools themselves are contributing to this. So if you look at the CDR, which we all have been focused on for many years, really one is in the early stages, one is really focusing on a very small area of change possible, because frankly, if somebody is pre-symptomatic, prodromal, early, there are really only so many sum of the boxes one can move in. And so within that category, what is even the reliability of saying whether a person has moved from a box or two in that scale? And I wanted to come back to a publication from many years ago that came from the ADCS, where the results were published from a validation or a training session of 82 raters. I was in the room during this testing, viewing the CDR videos trainings with John Morris, showing how he was meeting and testing the patient and their informant. And then the raters in the room would rate whether it was what they thought the CDR score was. And the bottom line is, if you go to the 0.5 level, the rating is about 50% accurate. That is, you might as well flip a coin to determine whether the person was 0.5 or not among this group. The bottom group is a group of people who essentially came from St. Louis, and I probably shouldn't say this, but I suspect actually had seen these videos before. In any event, this is a problem, and I think it's actually ignored quite a bit. On the other end of the spectrum, though, we still have the same difficulty of assessment. So, in later stage dementia, there are lots of behavioral problems. Actually, some of these can appear early on. So this is some recent data published by from the Dutch group of 1,000 people. All these individuals with Alzheimer's disease had amyloid PET positive scans, and just showing over several years, the differences, again, spaghetti plots of NPI ratings over time. And so being able to do, for example, a clinical trial where one is looking at symptomatic treatment and understanding whether you're having a benefit or not is a challenge because, again, of this difficulty in variability. The variability has a huge implication, and the fact that it's also compounded by the slow progression of disease. This is Ron Brookmeyer's multi-state modeling approach to looking at outcomes over time. I like this in the sense that you're talking about real state changes, not just potentially subtle changes. So it takes a long time to go from sort of asymptomatic amyloidosis to MCI. And if you look on the bottom table, these are sample size estimates that I've highlighted based on different assumptions of mortality risk and transition relative risks and so forth. But the bottom line is, if you look at these numbers, and if older people, you may have fewer subjects, but the bottom line again is thousands of subjects are needed in these trials. So there's really a critical need, I think, to show that we can actually detect meaningful change. The FDA has guidance documents about this recent or in the last few years, suggesting, in fact, that we need to go beyond the current metrics of standalone clinical uncertain standalone clinical meaningfulness, and that ideally the outcome measure is quoted here using the stage of disease, that is the early pre-symptomatic or prodromal stages in particular, will provide an assessment of meaningful cognitive function. They go on to further state that development of novel approaches to the integrated evaluation of subtle early Alzheimer's disease or pre-dementia, that these functional deficits and impact, that they arise from early cognitive impairment. So the current methodology assessment paradigm, we all know very well. It's what we all do or are used to doing. These are visits, baseline visits, and then follow-ups at a relatively brief period of time. They're episodically spaced, largely clinic-based. Much of the information is subjective that's obtained. They're obtrusive by their nature, and they're not very convenient. And this model has been going on for, well, since the beginning, I guess, when clinics were open. We've been modifying this recently through the pandemic because of the ability to provide a remote video or telephone assessment. And telephone assessments actually been around for a very long time, which is certainly helpful, but again, still has this episodic, brief, and limited ability to collect information. So one way to possibly, and you're probably not going to be surprised by this, to potentially change this is to use the technologies of, or the concept of pervasive computing and digital assessment. So using digital tools to provide data that comes from the home wirelessly, ideally that's by its nature objective. It's able to be provided continuously in real time. Ideally, it's an ambient kind of assessment, so it reflects what's happening in the home or in the community, and thus is ecologically valid. One can look at multiple domains at the same time, a holistic picture, because everything is time-stamped, so it's inherently aligned in that way. One is not necessarily having to rely on cognitive tests, but the cognitive tests are things like being able to remember to take your medications, or driving, or using a computer. These are cognitive tests themselves, and so we can look at those. And then the data itself is of the high dimensional nature, which allows one to look at more detail of intra-individual change. And this latter point in particular has major implications because it may, in fact, allow one to reduce substantially the sample sizes or the observation periods needed in a trial. So this table or this bar graph on the right is a redrawing of data from Hiroko Dodge's estimates of sample sizes needed that might be gained by using these digital methodologies, which I'll describe in some detail shortly. The large bar over on the left is an estimate of sample needed using the modified PAC, a traditional composite measure in a primary or secondary prevention kind of trial scenario. And as you can see, the much smaller sample sizes needed using digitally acquired things such as walking speed, total sleep time, computer use time, and a composite of all of those. So these are quite remarkable, and I know we might want to talk further, and we'll talk further about those in a moment. One of the other things to consider about these technologies is they may actually, they can facilitate more remote outreach and thus can increase inclusiveness. And so there are people who are shut out of participating in trials because they simply can't get to a trial. They don't live near a center. They're not able to make those trips, or they don't actually want to come in, even if they can make those trips. So there are many ways to capture this kind of data, and that's what I'm going to turn to now. There's hundreds of wearables. There's thousands of apps and many smartphones and other devices to deliver those apps on. There's a whole world of devices that are passive sensors, so passive motion sensors, or devices like electronic pillboxes that can capture this kind of data. And then there's an integrated version of all of these kinds of potential technologies, a so-called Internet of Things approach. So there are many ways to capture these kinds of digital biomarkers, is what they have been commonly called now. And I will ask the question, so what do you do? Is this something that you just kind of dive into? And what I like to do is approach this first by kind of talking about what not to do, or what some of the problems have been in using these technologies in clinical studies and clinical trials. So first of all, there's a huge cottage industry in developing computerized or digitized cognitive tests. Some of these are taking existing tests and just porting them to a digital interface. Some are developing more novel tests. But in the end, I would argue that these are still not, they're still have some of the same difficulties. They're episodically presented, they're not engaging, they're typically unidimensional, and they're not ecologically valid. And so these are, I think these can be certainly helpful, particularly for things like screening. So for example, just as some examples, digital clock drawing has been around for some time, and there's actually a lot of versions of this that one can look at both the traditional measures as well as some meta aspects, as well as things such as a touchscreen trails test shown here, where again, you can get the conventional measure as well as some other interesting aspects, such as the time it takes going from one target to the next, so-called thinking time or hesitation in thinking time. There are many issues with regard to using smartphones in general and apps in general. This study was a analysis of 8 Apple Research Kit based remote studies. These were done not in dementia, but in a variety of disorders, MS, asthma, Parkinson's, sleep, and so forth. And the bottom line here, though, is that despite thousands, tens of thousands of people initially signing on to these various studies, across the board, over 50% of the cohorts ceased engagement after 12 days. So not a great record in that regard. The other thing that, if you look more deeply at some of this interesting data from this paper, there was a very interesting disparity in terms of where the people came from who signed on over the internet. So there were other issues like this that need to be considered. One of the other pieces to this, in which may actually impact the usability, or the adherence and engagement, is the usability up front is often poorly considered. This is a graphic on the right of where people might carry their phones, and as you can see, women's clothing may not have the right pockets, for example. And then, just in general, I think we often don't think about what's happening in the world of our patients and people that we see. So this is a couple, perhaps a couple, or patients on the right of mine. If you look very closely, you can see that I'm on his laptop doing a video visit, but there's a few distractions here, and so and it turns out this gentleman was deaf from birth and had a cochlear implant, and there are lots of other issues going on. So there are lots of these challenges in these kinds of assessments. I think that, to summarize a lot of data and potential issues, there is no one simple digital tool or tools that should be engaged. It really needs to be focused on the old story of the right tool for the right job, and really understanding what is the proper outcome measures that you are needing for a particular trial. To try to get at having a way of providing these tools to the research community, back in 2016, 17, the NIH and the VA issued an RFA to create a general research platform for aging research that would be able to be used by multiple research teams, not necessarily just for dementia, but many aging-related functional areas. And so what you see around this home here is not technologies, but areas of great interest and need as outcomes in clinical studies for aging. And clearly, these are all very relevant to dementia. And so through this particular call, which we were fortunate to be funded for with some key collaborators at several other universities, we were able to build on a platform that we had already been working on through Orcatech, or the Oregon Center for Aging Technology. And so this technology agnostic, because we are not focused on a single particular methodology or device, so it needs to be open, shareable, and available, was created. The platform has to obviously engage conventional assessments, the things that we already do, bring in, if needed, EMR and other external data, and even data from the external environment. So even things like weather or air quality, things like that. But the focus has been on the home-based assessment of these areas of function. And then behind the door, if you will, of these areas of function are a toolbox of different kinds of devices and sensors that can provide information in these domains. And so these are examples. There are different, obviously a computer can be a laptop, a PC, a tablet, a phone, smartphone. It could be, a different operating systems, but they all can be used to look at aspects of cognition, and I'll expand on that shortly. Looking at mobility with motion activity, passive IR sensing in particular, bed mats for sleep, for example, but actually a wearable can also do that, or even passive sensors. And electronic pill box to look at medication taking and other behaviors, driving, physiologic function by a weight scale, by impedance scale, but also you could use a blood pressure monitor or other monitors, and even sensors that look at the indoor environment, such as the temperature, humidity, and so forth. All of this is importantly in the context of realizing that one will always have new sensors and methods that come along. So we've tried to develop the system so that it would be forward thinking. You can't build something that's future proof, but hopefully forward thinking and encompassing new things. The data is all, there's an extensive backend to securely capture this data and make it available for research. And I just put this up to call out Thomas Riley, who's sort of the genius behind the back end and the system, that there's a very extensive system involved to collecting and storing and securing and maintaining this system. There's some other details that are important. We focus on being as unobtrusive as possible. So most of these sensors are inobtrusive. Sorry, that keeps coming up not as well as I'd like. And then there's wearables and a bed mat. So there have been a number of studies over the years to look at specific aspects of function. And I'm just going to highlight a few very quickly here to for those who haven't seen this. I know some of you have seen me present these things before. So for everyday cognition, for example, one can look at computer use, measuring just the time that someone spends on the computer in a day or the days of the week, or the variability of that. And this has been shown to decline over time in people with mild cognitive impairment compared to age-matched normals. Mobility can be measured passively using sensors embedded in the walls, the ceilings, and this has been shown that walking speed and the variability in particular over time changes with MCI. Social engagement can be looked at in a number of ways. A simple measure that we like is simply the time together, time apart, time out of home, shown on this spiral plot, the black being the time to out of the home, red is time together, mostly at night. You can see that on this spiral plot over, I think this was a 30-day time period. Sleep, another core function for many reasons. Differences are seen here also in many different studies using either passive sensing, bed mat, wearable, and even more recently, a lot of interest in looking at the co-sleeping or the dyadic changes of how one person's sleep may affect another among people with or without dementia. Outside of the home, aside from just being out of the home, there are also a lot of activity. So for example, one can look at driving using a sensor on the data port. There are actually many of these commercially available. And this is just showing some data showing that people with MCI have some subtle but clear differences from those who are driving but don't have MCI. And then we've done some work with our colleagues in Minnesota, University of Minnesota and the VA there, A.G. Seeley, looking at this across states. And it's clearly a sensitive measure. One other important aspect of this is that all data can't be acquired passively. There are certain things you just have to ask. And this is something I think has been quite ignored in this literature of using devices such as wearables or phones, that the context that things are collected in really makes a difference. So internal states, people just have to report them. So is a person feeling down and blue? Are they lonely? Do they have pain? These can clearly affect what's happening. So we've developed over many years now a system where we actually ask people once a week through an old-fashioned email we push on Monday morning, these questions, force-choice questions about these key functions that could change behavior. And then this can be used to annotate and is used to annotate the database. And just as an example of that, quite dramatic, some early data from the pandemic, just showing, asking this weekly questionnaire prior to the pandemic. Then you can see in Oregon, in a group of people in the CART group, low-income housing, older adults, the increase in those reporting loneliness. And then in the bottom panel, a group of individuals in the minority aging research study that Lisa Barnes heads, looking at just step counts pre and post or after the pandemic was sort of announced, if you will. The other aspect to this kind of assessment, particularly online or using phones or tablets or PCs, is there's a lot of metadata that can be captured in the background. So whether it's how a person taps or swipes or moves their mouse, or the time of day that somebody actually typically takes a questionnaire, completes a questionnaire, or not so much the absolute result on a online screening test, such as a smart test, grid test, but the actual numbers of clicks, for example, that are used, it can be very powerful outside of just the absolute values. Very important also to consider validations. This is yeoman's work. It's not sexy. It's hard to actually get funded to do these kinds of studies, but they're very important. So for example, then there are many kinds. So there's a sort of issue where many of the research grade kinds of devices or actigraphs can be very helpful because of the data they can provide, but they're not very user-friendly. And so one needs to, for example, cross-validate these devices. So that's what these two panels are just showing, a study done to cross-validate a research-grade actigraph with a commercial watch actigraph, if you will. Another kind of validation is looking at the relationship of the digital or everyday cognition kind of tests versus the conventional tests. So the top table is just showing some data related to various measures of walking or mobility and the cognitive function scores. This was actually a normal sample. And then the bottom table is looking at weekly survey, online survey, adherence, time of completion, start of the time day, start date or start time of day, and the FAQ functional measure. There's also this issue of digital biologic, if you will, that is, does the measure relate to something in the brain? And so you're looking at volumetric MRI changes in medial temporal lobes are highly correlated with the change in the time on computer that I showed you briefly before. And then finally, a very hard to come by area is what I'm calling here a digital pathologic correlation, looking at the relationship of these measures. So time on computer, total sleep time, walking speed, and time out of home related to either plaque density or score or Brock score. Here, these graphs are Brock score, but there's a clear relationship of these to these digital biomarkers. So this system has been taken up by many collaborators at different levels. The red boxes are the original 4 CART sites that I mentioned that helped further tune and hone the system. I took the liberty of putting in orange the ADCS and the ADRC at UCSD because I'm sure we're going to have an increasing collaboration with all of my friends there, hopefully. But now I want to turn to some examples, particularly used in the idea or the context of trials. So the first thing I'll say is no drug trials have used this system. To recapitulate, we think there's a lot of potential value here because of, hopefully I've convinced some that there is the opportunity to get some, what I'll just call better data, but also to have major impact on the sample sizes, actually reducing the need to expose people to potential harm is the upside of reducing sample size need as well, not just cost. And the ability to make intra-individual predictions is also, I think, something very unique that also can be afforded by these kinds of data. So let me just turn to some of these examples in the remaining time I have, and I'm going to just go through these quickly to give a feeling for this. These are all non-pharmacologic interventions. So the first is the Iconx study that Hiroko Dodge has led these studies now over several years. The paradigm is to increase social engagement. And how do you increase the dose of social engagement to improve cognition and actually social function in people with MCI? You try to, in the concept here is increase a number of conversations or video interactions on a daily basis. So 4 days a week, people who enroll in these studies will have a conversation with a trained conversationalist on a device that Orcatech has made available to everybody. You don't need to know how to use a computer. It's a touchscreen enabled device. It's really a Chromebook. While that's happening, these conversations are recorded and the video is also recorded for later analysis, speech and language analysis. And a subset are using electronic pillbox as a measure of functional change. And there is also some MRI studies as well, but I don't have time to go into that. But bottom line is that this is a trial that's currently been, it's sort of second generation. The first generation pilot studies showed that people with MCI generated just a greater proportion of words compared to their conversationalists. There's a lot of potential reasons for that. And there was some interesting predictive value just looking alone at the conversational analysis. The figure on the bottom right shows just looking at actual types of words spoken. This is highly preliminary. I wouldn't go walk around saying that people with MCI say more swear words, for example. But there are these interesting signals that are being followed up in the study, the RCT that's going on now for iConnect. Another study has the acronym Stella. This is being conducted by Alison Lindauer, who's shown in the upper right on her screen. This is a behavioral intervention of individuals or caregivers of an individual with difficult behaviors living at home. So this is an entirely remote study. The platform is installed in the couple's homes, and the outcome measures are, aside from the typical caregiver outcome measures, also looking at whether there's improvements in sleep and socialization and time together and apart. Another example of a completed trial is this, what we've called the AIM study. This ambient independence, these measures to guide care transitions, or to reduce actually the likelihood of somebody transitioning to needing higher levels of care. So the idea here was that using a model of several of these digital outcome measures, we could predict the likelihood of somebody transitioning to a higher level of care. What we wanted to do was provide these digital biomarkers information to the care providing community in retirement settings, so CCRCs predominantly, and see if we could reduce the transitions to higher levels of care. And so a lot of work, a lot of background here, but we went through a lot of iterations of dashboards to present to the providers. This is just one example. It actually wasn't the one that was used in the ultimate trial. It was conducted at 7 communities, or 95 residents, 25 staff members, and they were randomized to being able to look at this data or usual care. And the outcome measure was the numbers of transitions to higher levels of care. And the bottom line is the technology worked terrifically and provided lots of good data, but the providers actually didn't look at the data very much. And we knew this because we could actually, down to the seconds of at least time they had the dashboards open and number of times they looked at them. So we still think that this is an unproven, that providing this information is helpful or not. We don't know that yet. I'm going to check my time here. I want to leave some time for discussion, so I'm going to quickly go through the next 3 things and then we'll finish up. This is just a study of art therapy and socialization. So these were people went to a studio, art studio actually, to participate in this program several times a week. And they had in their homes the platform that I described earlier, with some of the major outcome measures: computer use, or time on the computer, to outside of home, phone use in this case, and their mobility. And these people had a wide range of, for example, sort of social isolation before they entered the study. Results are shown here in a summary form using sort of heat maps that show the probability of these changes happening pre and post intervention. That's shown in the start, is the start of the intervention and at the end of the intervention. Then the timeline goes from left to right, and you can see just graphically that there was increases in, there were changes in on the UCLA loneliness scale, statistically significant. There's about 2 points. What does that mean? Hard to translate. But there was also increased time at home, number of computer sessions, walking or mobility changes, and phone calls after the intervention. The last study I'm going to just want to mention is this SHARP study. It's an acronym: Sharing History Through Active Reminiscence and Photoimagery. It's a really terrific intervention that Raina Croft has designed and run now, and keeps refining. The idea is to engage a group of African-American older adults in the community who live in gentrified neighborhoods to get out and walk and be physically active and socially engaged. So people walk in triads, and they carry a phone that has been pre-loaded with directions for each walk. They do this 3 times a week. And as they walk along, the phone will then show there's a area in their community that is of interest from their past. They stop, they discuss it, it's actually recorded, and then they move on. And they do this for, depending on which study they've been in, SHARP study, up to 6 months. In the latest iteration, the one of the triad has MCI, and the individuals will have the technology in their home to look at the outcomes as to whether they have reductions in weight, BMI, socialization, better sleep, and more physical activity outside of just the walk during the week. And this has been widely acclaimed and accepted. And Raina now is working with communities in Oakland and Chicago and Seattle and Atlanta to adopt and expand this intervention. I'm going to skip this for now, and I want to just end this tour of examples of how these technologies can be used in trials to go to the other end of the spectrum. So we've talked almost exclusively about people who have, early or prodromal MCI or Alzheimer's, but there's a terrible need for interventions for the later stages of dementia. There really are few effective treatments, and yet we still need to keep trying. So what you see in this graphic is a patient's room in a memory care unit, which is essentially two rooms. There's their room and then there's a bathroom. And it's been outfitted with essentially the same platform, but tuned to this purpose. So the motion sensors, there's one that goes over the bed, one that goes over, labeled here as a futon, or sometimes it's a couch, that are field restricted. So we get more focused data that's happening by location in the room. And then there are also environment sensors that I'll get to that look at things like humidity, temperature, light, and noise. And each person does wear a actigraph, and we use a bed mat to look at nighttime activity. This is some data from one of the patients who, this patient turned out to have frontotemporal dementia. Interestingly, it's one of the first ones when the system was put in place. Michael, our postdoc, actually, he's actually now assistant professor, he was concerned because he thought that technology wasn't working because there was no activity, but it turned out that the person was overdosed with quetiapine in the facility. They had made an honest error, and they were very, very apologetic, and they changed the dose. And what you can see here is the red lines are actual periods of reported, the vertical lines of agitation. And you can see this journey of changes in actigraphy counts, literal, just raw data over the life course of this individual's experience, really toward the end of his life. At the end of this graph, the person had died. And I think, though, it does speak to, the interesting question of, can we look more sensitively at responses to these medications that we use? Another way of looking at this here shown in a different patient. So this is focused on nighttime behavior and divided into agitated nights and non-agitated nights. And the agitated nights were specifically defined as when the person had received PRN medication for agitation. So somebody thought there was a problem. And you can, without statistics, you can clearly see these differences. In this case, particularly, there's a lot of movement out of the bed, not as much time in bed, and movement about the life space of this individual. And then the last thing I would point out is, this has been recently published as well, that there were very interesting and important relationships found between the agitated periods and these environmental cues, or well, we're calling them cues. So things like the sound level, the light, and the humidity, it turns out, seems to be quite potentially quite important as well, and maybe related to showering activity, as it turns out. So a lot to do there. I'm going to end now just by giving a few thoughts about things that we're thinking about on the horizon. So there's many, many opportunities here. I mean, it's really, I have to say, I am so fortunate to be able to work in this area and to work with an amazing group of people and colleagues. So I think there's lots of opportunity to develop new trial paradigms using these tools. There's lots of opportunities that I'm not going to go over all of these and just list them here. I'll just give a couple of examples. So this was just presented at AIC. You can go online and see this and hear me talk about this poster. But I wanted to just mention that one of the things that the pandemic did was push us to deploying the system remotely without having to step into the residence physically, the person's home physically. So we now have been able to send the devices, sensors in a box to the home, and then using little YouTube videos as well as online video assistance, or telephone assistance with one of our trained technicians. We've been able to successfully install this system. And I must say, I personally was skeptical of this, but we've done this now 21 times, and it really seems quite possible. I'm not going to go out on a limb and say everybody can do this, it's a piece of cake, but it's absolutely not out of the question that one could do a trial entirely remotely with this system, I think, at this point. Another area that I think is really ripe for changing how we think about things is to look more in terms of what people's habitual patterns are. So when you think about functional change, in fact, we focus a lot, obviously, on cognition, but unless you have functional change, you're not demented by definition. So what is the change in function really? What are the signals that might be changing early? And really, one of the things that's common about human behavior is we're creatures of habit. So if one could detect how these patterns that we all have change over time, that may be a signal that's very important. So what you're seeing here this is work of Chayi Wu, who has been mapping the sort of habitual patterns of activity across the day, looking at things like room transitions and dwell times, or on the right, average hourly room occupancy. So you're looking at the, as these dots go and come, you're looking at the changes in those patterns. And if you summarize this data as she's done here, and this was also presented at AIC, so you can go online and look at her presentation. What was found in this preliminary data is that people with MCI seem to be less likely to leave home for prolonged time in the morning. They're much more likely to leave home in the afternoon. And they spend more time in the kitchen and make trips to the kitchen in the evening than age-matched comparison volunteers. So we're very interested in this, kind of exploring this further, and also looking at the interactions of couples in that regard as well. And then finally, something that's particularly near and dear to us here in the Northwest, and I know in California, actually out in the West in general, is air quality. So this is data from last year, where what you're seeing in the left panel is the air quality reported. So this is outdoor air quality reported over time. It's a little frozen, but that's okay. One of the interesting opportunities, though, is to now look at this outdoor air quality, which is freely available by public databases, but also looking at what's happening indoors as well, and the effects that may have on people as they not just age, but also even in clinical trials and studies. So with that, I'm going to just summarize and say there's a lot of things that we can do. I didn't talk very much at all, really didn't talk at all about using digital tools and methodologies for the front end of recruitment and identifying people. There's the phase of trial enrichment. I mean, much of this is really important in terms of knowing what the targets are, and knowing the group you're going to enroll into a study. But I think the place of important engagement of technology is in what I call behavioral phenotype enrichment. That is, what we know is you might identify people who are amyloid positive or tau positive, or ApoE positive, ApoE4 positive, but you don't know as much about how they might progress in a trial. And so you can fairly quickly, using the digital behavioral phenotyping, determine those that might be progressing more rapidly than others, for example, in particular domains. The technology, once it's installed and deployed, continues on into the trial proper. And then you can do the assessments that I described earlier this morning in my talk, and really look at individual rates and changes of progression depending on treatments that have been applied. So with that, I'm going to thank you all. And I also am going to give a plea to collaborate. I really think that this is the kind of thing that can't be done by one group. The CART idea was to see this platform and these methodologies used more widely, and hopefully that will happen soon, or is happening, but will happen even more broadly.